Now, hemoglobin was the molecule that bound oxygen, the main driver, the main carrier of oxygen in the blood. It's the thing that allows us to carry oxygen in the blood. Remember, oxygen does not dissolve in blood easily. Very low solubility coefficient. Hemoglobin is the only thing that allows us to transport oxygen in the quantities required. And we left before the break introducing this figure, the hemoglobin oxygen association curve. This simply shows you how full the hemoglobin is for a given PO2. And there's a few candidates on here that you might recognize. PO2 of 100 is around what you see in the alveoli, in freshly oxygenated arterial blood. And so hemoglobin is very saturated, nearly 100% saturated. And as you get down to the tissues where PO2 drops, saturation also drops, but not very much. Only about a quarter of the oxygen carried in the blood is used in normal situations. So the PO2 here of about 40 at the tissue, still 75% saturated, which is great. That means that we have reserve capacity. We can tap into and extract more oxygen if we need to. For instance, in a situation like exercise. That extraction might be shown here if PO2 were ever really low, 20 millimeters of mercury. We could reduce the amount of hemoglobin bound with oxygen to 35%. So 25% is used. It's fully saturated above 60 millimeters of mercury. We're very resistant to changes in saturation in arterial blood, which is good. We want it to be fully saturated. And now we're moving forward to understand how this changes. Because this isn't static. This isn't static. It varies depending on internal conditions. It varies depending on which part of the body you're in. Because not all of these elements are consistent from location to location within the body. So factors that affect the affinity of hemoglobin for oxygen. And the affinity, this is the idea that hemoglobin attracts oxygen, or the likelihood that hemoglobin will bind oxygen. If there is a high affinity, hemoglobin will very likely bind oxygen. It will do it easily. It will readily bind oxygen. If hemoglobin has a low affinity, it's hard to bind oxygen. So affinity describes how likely hemoglobin is to bind oxygen. And the first item on this list we've already explored in detail. If the PO2 changes, hemoglobin's affinity for oxygen also changes. High PO2, high affinity. We're over here on the right-hand side of the graph. Low PO2, lower affinity. We move towards the left-hand side of the graph. And while the first example that I showed you only depicted the black line in the middle, the relationship was fixed and static, and you slid up or down depending on what PO2 was in the immediate surroundings. The relationship can shift left or right with changes in pH, changes in temperature, changes in bisphosphoglycerate. Bisphosphoglycerate, which I'm only abbreviating as BPG. We'll talk about it. It's coming up. It's a metabolite. Bisphosphoglycerate. These things can affect the affinity by pushing the curve to the right or pushing the curve to the left. So let me ask you, if one of these variables or factors increased the affinity of hemoglobin for oxygen, made it more likely that hemoglobin was going to bind oxygen, would it shift it to the right or the left? Left is correct. Great answer. If one of these stimuli increases the affinity for hemoglobin, that means that at any given PO2, more oxygen is bound. So in this case, the blue trace is shifted to the left. If I look at a PO2 of 30, in a normal situation, I might be, 45% saturated. But if one of these elements increases the affinity, it's shifted to the left, and I'm now binding 65%. 65% of my hemoglobin is bound with oxygen. The affinity is increased. And the reverse is true as well. If one of these factors decreases affinity, it shifts it to the right. The red curve that's depressed on the right-hand side. So this is very general terms. Let's talk specifics. pH is a fantastic first example. And it is the example that I used on that first page, just with all the labels removed. pH can affect whether or not hemoglobin will bind oxygen, will affect the affinity of hemoglobin for oxygen. Now I'm just going to tell you right away that an increase in acidity will decrease the affinity of hemoglobin for oxygen. Higher acidity, a more acidic environment means that less oxygen is bound. Less oxygen is bound, that means the curve shifts to the right. It's depressed. At any given PO2, saturation is lower. Ignore the gray thing for now. I just wanted to bring up the relationship. When I say acidity and you're looking at pH on the graph, I know we're not a chemistry class, but we should be able to wrap our heads around this concept. If acidity is high, something being acidic simply means that the concentration of protons is large. And the scale that we use to describe the concentration of protons is pH. Just like the scale that we use to describe heat is a temperature scale, we use pH to say whether or not the concentration of protons is high or low. But it's a little bit twisted. When pH is a small number, acidity is high. It's not intuitive. When pH is a small number, proton concentration is high, acidity is high. And vice versa. When pH is a high number, acidity is low. You are more basic. The concentration of protons is lower. So when I say that high acidity decreases the affinity for oxygen, I'm looking at this specific case. Low blood pH, here a pH of 7.2, is more acidic than normal. It's not a big change, but it is a change. pH goes down, which means that acidity goes up, and at any given PO2, the amount of oxygen bound to hemoglobin is lower. The affinity is lower. You might think this is a bad thing, but consider where in the body you would really run into this acidic environment. Any thoughts on where in the body you might run into this acidic environment, or in what situations? I hear murmurs over here. What environment? Any ideas? Pardon me? Stomach is a place where there is an acidic environment. Are red blood cells and hemoglobin apparent in the stomach? No. Good idea, though. That's fresh in your minds after lab this week and lab before the break. Stomach, acidic environment. But the blood, where would the blood be acidic? I always default, if you're ever posed with a question like this, it's never bad to guess exercise. I always default to the situation of exercise. Intense exercise can acidify the blood. So at the tissue, if you're cycling really hard or running really hard, the blood can be acidic. And it sounds, at first pass, it sounds bad to say less oxygen is bound, but when you reframe it and think, if the environment is acidic, it makes unloading of oxygen easier. Well, in a situation where I'm exercising and hemoglobin is at the tissue, I want to unload oxygen. That's a good thing. Acidity, in this case, helps to unload oxygen. Similarly, a change in PCO2 will cause a similar shifting of this equation. And we're going to see today how PCO2 and acidity are almost linearly related, almost one-to-one. If one goes up, the other goes up, and vice versa. PCO2, or the amount of CO2 in the blood, has an immediate proportional effect on the acidity of the blood. We're going to see that today. So if PCO2 goes up, the blood becomes more acidic, and the affinity is decreased. High PCO2 decreases the affinity of hemoglobin for oxygen. This also helps at the tissue in a situation of exercise or otherwise. Right? The tissue is what produces CO2. Refer back to that diagram, the cardiovascular circuit, where we had O2 and PCO2 throughout the arterial and venous circulation. Where does PCO2 go up? At the tissue. Well, if PCO2 goes up at the tissue, that helps to unload oxygen. Again, that's a good thing. So acidity, PCO2, two factors that will shift this relationship. The opposite is true as well. If the body becomes more basic, or if you hyperventilate and PCO2 drops, you make it less likely that hemoglobin will release oxygen. Another situation that, again, also happens to coincide with exercise is a change in temperature will affect the ability of hemoglobin to bind oxygen. Now, the last two examples were a somewhat chemical stimulus, and we're going to go over the mechanisms today, how pH or PCO2 push oxygen around. Temperature is not a chemical removal of oxygen. Temperature simply describes the shape of hemoglobin. Hemoglobin is a protein. Shapes of proteins change with temperature. As temperature goes up, they get more labile. They move more. They expand. As proteins get colder, the opposite happens. They get stiff. And the sites of binding close off. So in an environment that's hotter, where the sites for binding oxygen are more open, it's easier to release and offload oxygen. This also happens at the tissue during exercise. Another factor which helps to unload oxygen where it's needed. Increased temperature decreases the affinity of hemoglobin for oxygen. This is perhaps a somewhat difficult concept to understand. But think of the proteins aren't fixed. They're not Lego blocks. They're very fluid. They move around. They twist. Their shape changes. And if they become too hot, more than 43 degrees, if they become extremely hot, they can unravel completely. We call that the process of denaturing proteins. You see it all the time when you cook eggs. We don't want that to happen, and that's not what I'm describing here. They just simply loosen a little bit, and then binding oxygen becomes harder. I realized there is not a graph, a figure representation of what happens with bisphosphoglycerate, BPG. So I'm going to ask you. I'll tell you a couple things about it first, then I'll ask you what you think happens. BPG is produced during normal metabolism. If I'm at the muscle and I'm hemoglobin and I'm going to react to BPG, do I want to make it easier to unload oxygen when there's a lot of metabolism happening? Or do I want to make it harder? Who wants to take a guess? BPG indicates metabolism is happening. Should that help to unload oxygen or not? I think all the way back to the end of first semester when we talked about metabolism. We use oxygen to make energy. That's a pretty big hint. You guys have it? Anyone? No? Well, if there's a lot of metabolism happening in the vicinity, it probably means the demand of oxygen is really high. I'd really like to unload oxygen in that situation, make it available for metabolism. So like temperature, like a high acidity, like high PCO2, a high BPG content will decrease the affinity for oxygen. Helps to unload it. A different take on this idea that was listed on the first slide on the list of bullet points is shown here. The type of hemoglobin will affect its affinity for oxygen. Normally, hemoglobin is hemoglobin in adults. It doesn't really change. Red blood cells are processed and broken down, and heme is recycled. The proteins are recycled. Amino acids are used for other proteins in the body. But hemoglobin structurally is consistent in you over time. But where there are different types of hemoglobin, one of them is shown here, in the fetus versus the mother. And this is really important to ensure transfer of oxygen from the mother to the fetus. What you're looking at here is a representative trace. Fetal hemoglobin shown on the dashed lines. Maternal or normal adult hemoglobin is the solid red line. And at any given PO2, fetal hemoglobin is more saturated. That is, at any given PO2, fetal hemoglobin has a higher affinity for oxygen. It more easily binds oxygen. If it didn't have an altered affinity, oxygen would never move from the adult hemoglobin to the fetal hemoglobin. It's because of this separation that you can oxygenate fetal hemoglobin at the placenta and then deliver it to the fetus proper. At any given PO2, fetal hemoglobin has a higher affinity. This ensures oxygen transport. I'm going to show you a slightly different variation or a slightly different representation of the exact same information with some new stuff shown here because I think it's really important to continue passing the baton. And that will become clear in a second. So this is the same information that we've just been talking about. Hemoglobin O2 association curve, and the normal adult curve is shown here in green this time. Chose green for this figure. The concept that we just discussed, fetal hemoglobin, shown here in blue. Again, at any PO2, fetal hemoglobin has a higher affinity for oxygen. That ensures transfer from the mother to the fetus. And we've got another red line here that I'll talk about in a second. But first, I want to just make sure that you're aware that this is the exact same information that we just saw. Next, There's a new term that's introduced on this slide. It's a way to describe how strongly hemoglobin binds oxygen. That is the P50. The P50. And the P50 is simply what partial pressure gives me 50% saturation. P50. What partial pressure gives me 50% saturation? A higher P50 means a lower affinity for oxygen. A higher P50 means a lower affinity for oxygen. Let's look at the adult hemoglobin. Their P50 is 27. So at a PO2 of 27, half of the hemoglobin is saturated, half is empty, half is not binding oxygen. P50 for fetal hemoglobin, 19. So at a lower PO2, we observe 50% saturation at a lower PO2. This is the same concept or the same idea as we've discussed at any given PO2, which curve is higher, which one has a higher affinity. This is just reframed slightly. So I don't want you to be confused what that number means on the graph. I only present this graph because as we follow the flow of oxygen in from the environment, the handoff between the alveoli and the blood, pretty straightforward. Oxygen moves in, red blood cells are right there, hemoglobin is right there, and it binds it quickly. And it binds it throughout the entirety of its travels in the arterial system. And we know that it's unloaded at the tissues. We know it leaves hemoglobin and then something happens. But what we don't know is what happens to describe that something. Oxygen has to go from the red blood cell into the tissue, through the tissue, and eventually down to the mitochondria. Remember back to the metabolism section. But there's a problem. Cells are full of water. They're full of fluid. And oxygen doesn't dissolve in body fluids very easily. So, how do we ever hand off oxygen from the red blood cell in the blood, in the arterial system, to the mitochondria? There's a big gap. And that gap is filled by myoglobin. Myoglobin is a very specific variant of hemoglobin that's only found in muscle. And if you follow the logic of this P50, or if you follow the logic of at any PO2, which one has a higher saturation, myoglobin is almost always completely saturated with oxygen. It's near 100% saturated at anything over about 20 or 25. So, analogous to how fetal hemoglobin will take or has a higher affinity that allows the handoff of oxygen between the adult and fetal hemoglobin, myoglobin, having a much higher affinity, allows the handoff from the arterial blood into the cell. And only when PO2 gets into the single digits does myoglobin release oxygen. So it's a really strong oxygen magnet. Anywhere in the cell, myoglobin binds oxygen. The only place that it will release it is where PO2 is in single digits, which happens to be at the mitochondria. The place that uses oxygen. You can think of it as like a well or a sinkhole or a drain. It's all being funneled to that one specific location. So myoglobin allows the handoff from hemoglobin in the blood to the mitochondria to allow aerobic metabolism to happen. A little preview of what's coming up. So does that make sense? Factors can affect affinity. On those graphs, if the line is higher, more oxygen is bound, higher affinity. If the line is lower, less oxygen is bound, lower affinity. Oxygen is drawn to those structures that have higher affinity. So in effect, it moves up on any of those graphs. So we've completely covered transport of oxygen. We're effectively ignoring dissolved oxygen in the plasma because it is so small that it makes no physiological impact. There is little dissolved oxygen. We certainly cannot extract enough from the dissolved portion, and it's generally resistant to change, so we're not going to talk about it too much. Hemoglobin is what transports and delivers oxygen to the tissue. We've covered that in depth. What we're turning our attention to is CO2. Now this slide was presented earlier. I'm just revisiting it here. There are three ways that CO2 is transported in the blood. And the dissolved portion, it's still small, but it's a relatively larger influence than the dissolved portion of oxygen. So we are going to discuss that just briefly, but the two major routes of CO2 carrying in the blood are bound to hemoglobin, or bound to proteins, not just hemoglobin, and or through the carbonic anhydrase reaction shown here. The large majority is converted to bicarbonate by the carbonic anhydrase enzyme. This explains what I mentioned earlier, that CO2 and pH are tightly linked. This equation explains why that is. This is what we call a near equilibrium equation, which simply means if you have more substrates, it moves to the right and makes products. If you have more products, it moves to the left and makes substrates. It's always in perfect balance. If it's not, it moves so that it is in perfect balance. And if you imagine a situation where PCO2 goes up, if I have more substrates, it moves to the right to create products. What's one of those products? Hydrogen ions. We went through the painstaking description of what acidity is today, a measure of the concentration of hydrogen ions. If there's more CO2, there's more protons, CO2 increases acidity. That's by far the most important mechanism or route by which CO2 is carried in the blood. The other, or the second most important route, is bound to proteins. Here the protein is hemoglobin, but there are other proteins in the blood that CO2 can bind to. When CO2 binds to a protein, it makes a carb-amino complex. Carb for carbon dioxide, amino because amines are the base unit of proteins, carb-amino. If you were to draw it out and write the reaction, it would just look like this. Simple addition and subtraction. Hemoglobin plus CO2 makes HbCO2. Let's look at this in detail and start to explore how O2 and CO2 carrying are related. They're not independent. Let's explore them in a bit more detail. The example we'll go through in detail is at the alveoli. There's a second example on this slide, which is at the tissue. I'm not going through it in as much detail. I encourage you to do that. Often, it's just the reverse of what we're going to talk about at the lungs. So this top panel, this top example, is at the lungs, trying to oxygenate venous blood and trying to expel CO2 from venous blood, put it that way. Now, I will lead this description with a brief statement. If we're considering CO2 movement first, we know at the lungs we need to expel CO2. The gradient favors removal of CO2 from the blood. That is, it's higher in the blood than the alveoli, and it's higher in the alveoli than in the environment. And so as CO2 leaves the blood, it pulls CO2 out of those respective compartments that we just talked about. Pulls CO2 out of the plasma, pulls CO2 off of proteins, and here, what we're going to talk about in more depth, pulls CO2 through the carbonic anhydrase reaction. What's not immediately obvious is how this relates to the binding of oxygen, but we'll get there. So let's go through this deliberately. This is the alveolus over on the left, so that's the base unit of the lung, and we already know what the gas looks like in the alveolus. What's being expelled? CO2 is leaving the alveolus as we ventilate, and that creates a bit of a lack. If we are pushing CO2 out, it has to be refilled, and it's immediately refilled from the surrounding interstitial fluid. CO2 moves into the alveolus from the interstitial fluid, and the chain reaction continues. It moves across the capillary wall. That CO2 comes from the plasma. The plasma then needs to be replenished. CO2 leaves the red blood cell to enter the plasma, and CO2 in the red blood cell then needs to be replaced. What replaces free CO2 in the red blood cell? Those three mechanisms that we just talked about. Some CO2 is bound to hemoglobin or other proteins. If dissolved CO2 is removed, well, I'm going to break CO2 off of those proteins. It's going to fill the gap that was just created. I have CO2 being unloaded from those proteins. Where else can CO2 come from? CO2 can come from the carbonic anhydrase reaction. If there is not complete balance, this equation, this reaction will move so that there is balance. The situation I'm describing here is an imbalance where there's fewer, we're going to call those products. They're on the left-hand side. The left-hand side concentration is lower, so things are going to move to the left. H2CO3 breaks up into carbon dioxide and water, pulling bicarbonate and protons through the reaction till it's completely balanced. And that describes the process by which CO2 is removed. Removing from the alveoli creates a pull through all of those mechanisms, through all those compartments, by which CO2 moves into the lung. Now I'm in a situation where I've removed bicarbonate and protons, and so those are also going to tend to be replaced. Bicarbonate replaces any bicarbonate that was consumed by the reaction, and protons are replenished in a simplistic manner by hemoglobin. We don't fully understand how this works. Some of them come out of solution, but they have to come from somewhere because we're using them in this equation. You can think of the protons that aren't just floating around freely as placeholders. hemoglobin will bind some protons as a placeholder. Placeholder for what? Placeholder for oxygen. So HbO2 protons are removed, the placeholder is open, hemoglobin is open, allowing oxygen to bind. Pulling substrates through this reaction creates the ability to bind more oxygen. It opens up hemoglobin as a protein. Well, just in time, because in this environment where we're removing CO2, oxygen is also high. Oxygen then will move in down its concentration gradient. It will move across the capillary wall. It will move out of the alveolus into the interstitial fluid and be renewed by ventilation from the environment. So notice how as we expel CO2, we open up the sites of binding for oxygen, allowing the exchange of gases to occur. And that's why it's called gas exchange. No, there. Oxygen's coming in from the periphery. So you can think of it as CO2 is being expelled and it pulls everything through. You can think of it as O2 coming in from the environment pushes everything through. And they're both right. The point is O2 loads, CO2 unloads. All of these mechanisms are in place but reversed at the tissue. Gradients are reversed, direction of movement is reversed, the reactions are reversed. CO2 is loaded, O2 is unloaded. So go through the example and see if it makes sense that CO2 produced at the tissues combines with water in the carbonic anhydrase reaction, producing bicarbonate and protons, and how that might influence the binding of oxygen, or the lack thereof, perhaps. So, important concept, good to know the details. So this is a natural breakpoint. We'll come back and talk about the control of respiration in class on Thursday.